The Hershey and Chase blender experiment, as told by Sidney Barker. This experiment was conducted in 1952 in order to find out which part of a bacteriophage served as the genetic material that would be passed on to its progeny. The two theories at the time were proteins, which were accepted as the method of transmission because they are more diverse and complex than nitrogenous bases, which were thought to be. DNA was the other option and had already been established as the transmitting principle for genetic material in bacteria by Avery, MacLeod and McCarty in 1944. Bacteriophages are viruses that inject their genetic material into bacterial cells in order to reproduce. Using this information, Hershey and Chase used two different radioactive tracers to figure out which unit held the phage's genetic information. Because DNA contains phosphorus, radioactive P32 was used to trace it, and radioactive S35 was used to trace the proteins as amino acids contain sulfur. The two different phages were allowed to infect E. coli bacteria, and then the samples were spun in a blender to break apart the protein capsule. The samples were then spun in a centrifuge to separate out the cells from the proteins. The results of this process were phage protein capsules with the sulfur tracer, found with E. coli with no tracer, along with E. coli bacteria with a phosphorus tracer that were found with a capsule with no tracer. Since the DNA tracer was found inside the bacteria and not in the phage at the end of the experiment, it means that the DNA is the method of transmission of genetic information in bacteriophages.